is over, and that is on Blackboard. And the extra credit video or paper is due Monday, and if you've forgotten where that is, it's under the exams link, because I always kind of have all the credit to the exams. So that's where it is on Blackboard. And the course evaluation, the completion, don't email it to me, I will lose it, because later I'll see that you didn't bring one in, and I'll look at the email, and it turns out you didn't send it to me from a Whitworth, you sent it to me from some other thing, and then I can overlook it. So it's better if you upload it to Blackboard. You just click on the link with the instructions, and it'll take you to the place to upload it. Okay. Christmas party, yeah. Christmas party today at 3, 3rd floor. There's food, please come.
What kind of food?
I'm bringing meatballs. The free kind. So
I'm not planning on making dinner.
Well, I didn't eat breakfast because we're going, because I don't have any.
You made it?
I'm planning on eating a lot after.
part of it.
Okay.
I'm proceeding to practice at 4.
What's that show is?
It's just
Are you sticking around, right? Okay. I have to stay after.
She's going to do the video with us, aren't you?
We're going to write the video. Figure out what.
Write the video, film the video.
No, we're going to figure out what the heck to do with the video first.
Oh, right then.
We ended talking about pure non-competitive inhibition.
Breaking Bad.
That is what you observe when you have irreversible inhibitors. And irreversible inhibitors are often poisons. And so I just have a few examples of poisons because this is always fun stuff. So ricin is a glycosidase that modifies ribosome. So this poison is actually another enzyme. And so if it's a glycosidase, what is it doing? Okay, so cutting. You've seen other sidases like this before. What kind of enzyme is that usually? I think you've seen other sidases. Have you seen other sidases? Is it EC3? Does that help? No? Hydrolase, yay, okay, so it's a hydrolase. And then the glyco tells you what you're maybe cutting.
Glycosidic bonds.
A glycosidic bond, right. So now this is in ribosomes, and I know we haven't talked about ribosomes, but you might know something about those from biology. So what are ribosomes composed of?
Protein.
Okay, so there's some RNA, there's some proteins. Okay, there we go. So RNA and proteins, and glycosidic bonds. Where do you see glycosidic bonds in RNA?
Between the phosphate.
Yeah, that's the tricky thing. So phosphate backbone is all like phosphoester, phosphodiester. But how do you link your ribose up to your base? So glycosidic bond. Yeah, it's N-linked, but it's a glycosidic bond. So this enzyme goes and very specifically cuts off one adenine base in a really important region of one of your ribosomal RNA subunits, and it kills the ribosome. The ribosome will not work after that. So lethal dose is 22 micrograms per kilogram, which is a pretty potent toxin, and this is an assassin's poison. And so the very famous one is the umbrella assassination, if you've ever heard of that one. It was supposedly the KGB, and this is back Cold War, right? Where they assassinated a defected Bulgarian dissident who was saying bad things about Russia and the KGB. And he was killed. And the weird thing was that he was just walking along the street, and he was in London at the time, and someone poked him in the back of the foot with an umbrella that was just sharp enough to graze the skin. And it injected some of the ricin, and it was enough that 2 days later he died. But it takes a little while for that to affect all your cells. But there's an irreversible. So it covalently modifies your ribosome.
It's also in Breaking Bad.
Cyanide. Okay, so cyanide poisoning is something that most of you have heard of, and that was like, I didn't want to have cyanide as a grad question because it scared me. But this one reacts with an enzyme that's found in your electron transport chain. So again, in biology, you've seen this before. Great, and if not, then if you take Biochem 2, you'll see it next semester. But it affects cytochrome oxidase, C oxidase, which is this one right here. And it's kind of the final step of that electron transport chain. And what happens is that cyanide gets in there and it actually ends up coordinating some of these copper and iron centers, which are necessary to pass electrons through the transport chain. So if you've got this foreign thing that is messing up your iron centers, then you can't pass electrons. And so what that means is that you can breathe just fine. You can breathe in the oxygen, but since you aren't coupling reduction of oxygen and ATP synthesis anymore, because you have this disrupted electron transport chain, and you suffocate even though you can breathe. Sarin, so you know the first one was an example of an actual protein inhibitor, and then we have a small molecule inhibitor that affects metal centers, and sarin is a small molecule inhibitor that actually affects an enzyme by covalently modifying the active site serine. So this is a catalytic triad type enzyme. So this one affects choline esterase, which you need to recycle neurotransmitters at synapses. So without it, you can't recycle your neurotransmitters. And sarin is this lovely little electrophilic thing here. And so if you've got a hydrolase, which is what this esterase is, that needs to nucleophilically attack its substrate in order to break these ester bonds, it can attack serine instead, and then it covalently modifies that active site serine, and then it doesn't work anymore. And so this is a nerve weapon, weapon of mass destruction. These are all examples of irreversible inhibitors, and they do something to permanently change, usually through a covalent bond, or changing covalent bonds. Your enzyme.
That was the gas that they weren't sure which group in Syria was releasing.
Yeah. No, sarin is a really common gas. Everyone is scared that everybody else is sarin. Okay. So enzyme regulation. This is kind of the whole point of everything that we've been talking about with inhibitors. So enzyme regulation can be done by one of two approaches. One, you regulate the availability of the enzyme. Through expression and degradation. So basically you're regulating gene expression at this point. Or protein recycling. So how rapidly the proteins get degraded in a cell. This method is only good for long-term changes. Why would that be? Ideas? Why only for the long term? Really slow. Okay. It takes a little while for you to upregulate gene expression. It takes a little while for you to degrade the existing enzymes in a cell. And so because it's slow, comparatively, because remember our enzymes that are catalyzing reactions with turnover numbers that are powers of 10 in magnitude, compared to the catalysis that's happening, this is really, really slow. And so because of that, you only want to do it if you need to make a change for the long term. What typically happens on a faster time scale is you regulate activity directly. And that could be through binding of an effector, molecule, or sometimes these are called modulators. An inhibitor is a negative modulator, and an activator is a positive modulator. Or you regulate the activity through covalent modification of the enzyme. So post-translational modifications, we've talked about these before. What are some examples? Acetylation, is that what I heard? What else? Phosphorylation. What else? Sorry? Methylation. Okay. A little louder.
Carboxylation.
Sure, why not? Carboxylation. You can pick your favorite functional group and just put an ation at the end, and probably will be okay. Let's see, acetylation, phosphorylation, methylation, carboxylation. We talked about hydroxylation. I don't remember. Is that all of them? Probably not. But lots of different things that we can do to amino acid side chains to change how our enzyme functions. So, allosteric regulation is something that we need to define. This is the example where we have an effector. This is an effector that binds to the enzyme at a site other than the active site. Binding affects activity. So this could be inhibitors or activators, either one. So either direction. And your type of inhibition is going to be what? Uncompetitive, there we go, uncompetitive. Or mixed. Which is that other word that, I don't know which is the right word. I still haven't decided which is the, I think it's both that English actually. So mixed or non-competitive. So it could be either one of those, you don't really know. You should go into the lab and actually measure the kinetics to find out. But what it won't be is competitive because you're not binding at the active site where the substrate is binding. So it's going to be either competitive or uncompetitive or non-competitive. So let's see, an example that your book goes through is aspartate transcarbamylase. Also called ATCase. And this is part of the pathway. Synthesize basically C2P, or at least the C anyway. So it's to mix that Z. This enzyme is allosterically controlled. And it's allosterically inhibited by C2P. And the enzyme is allosterically activated by ATP. So ATCase, and this particular enzyme is at the first step for the synthesis of pyrimidines in general, and so that includes uracil. And this is a reaction that we will actually not talk about in the second semester. I skipped the whole nucleotide synthesis because we just don't have time. But it's a great example of allosteric regulation, and it's going to be a good example of feedback inhibition. So basically, a transcarbamylase, that kind of tells you what it's doing. So it's a transferase, and it's transferring a carbamyl